Hello students, let's study general mycology and dermatophytes. Let's study general mycology. Fungi are eukaryotic. They have rigid cell walls containing chitin, mannan and other polysaccharides. Their cytoplasmic membrane contains sterols. They possess two nuclei with nuclear membrane. They divide asexually, sexually or by both, maybe unicellular or multicellular. The classifications are of three types: morphological, taxonomical and pathogenic. So first, morphological classification. So in that, yeast is there. Example, Cryptococcus. Yeast-like cells, they are oval or round cells with buds. Example, Candida. Molds, so they have hypae, elongated structures. Example, dermatophytes, Aspergillus, Rhizopus, Mucor and Penicillium. Dimorphic fungi, so they have two forms at the 37 degrees Celsius and at the room temperature. So examples: Blastomyces dermatidis, Paracoccidiidis brasiliensis, Coccidiidis imitis, Histoplasma capsulatum, Sporothrix schenki and Penicillium marnifi. Second, taxonomical classification based on sporulation. So zygomycetes, sexual zygospores with sporangium within sporangium, asexual sporangiospores, they are endogenous. Example, mucor. Ascomycetes, sexual ascospores within the ascus, asexual conidia, exogenous. Aspergillus basidiomycetes, sexual basidospores in the basidium, asexual conidia, exogenous. Example, Cryptococcus. Deuteromycetes fungi imperfecti, sexual spores not known, asexual conidia, exogenous. Most clinically important spores belong to this group. Based on the infection, that is pathogenic classification, so there are superficial mycosis, superficial surfaces of the body. Example, dermatophytosis. Subcutaneous mycosis, subcutaneous tissue, dermis and subcutaneous tissue. Example, mycetoma, rhinosporidiosis. Deep fungal infections, disseminated. Example, histoplasmosis, blastomycosis. Opportunistic fungal infection, when the immunity is lowered, then fungal infections are caused. They are example, candidiasis, aspergillosis, zygomycosis, mucormycosis, cryptococcosis. Pathogenesis of fungal infection, source of fungus, environment, warm, humid area, air, plants and soil, animals, unsterile instruments and hospital equipments. Site of infection, CNS, eyes, lungs, bloodstream, skin, subcutaneous tissue. The fungal agents according to the sites of infection causes disease. So Cryptococcus is CNS, eyes, rhino-orbital mucormycosis, skin, dermatophytosis, lungs, aspergillosis, bloodstream, disseminated candida, coccidioides, Paracoccidioides systemic and mucocutaneous, subcutaneous histoplasma mycetoma. Laboratory methods for isolating and identifying clinically important fungi. So first, specimen collection. Second, direct demonstration of fungal elements by microscopy. Third, isolation of the fungus in culture. Four, immunological evidence of infection. Five, other tests for the diagnosis of fungal infection. Example, skin test for hypersensitivity, molecular methods. First is specimen collection. So according to the site, superficial infection, skin scraping, then hair plucked from the root and nails clippings. Subcutaneous tissue biopsy, mycetoma, granules from the infection or sinus tract, deep and opportunistic infection, blood, CSF, pus discharge, sputum and other body fluids and aspiration. Microscopy, second step, the potassium hydroxide preparation 10%, it digests keratin. So KOH wet mount can be done for demonstration of fungi. Second, gram staining. In that they are gram positive is cells can be seen. Example, Candida. Cauliflower white stain. Then India ink or nigrosin staining for capsule demonstration of the Cryptococcus. Methenamine silver stain and periodic acid shift stain can be seen in histopath preparation. Lactophenone cotton blue. LPCB can be seen for the demonstration of the colonies. Third step is culture, temperature ranging from 25 to 30 degrees Celsius, slow growing, and then media used, tabloids, dextrose are most common fungal culture media. It contains dextrose for nutrition to fungi and acidic pH to inhibit commensals. Second special media, selective media is bird seed agar, niger seed agar for Cryptococcus, cornmeal agar for the chromogen candida, blood agar, brain heart infusion agar. In preparation of slides, LPCP mounts, so taste mounts in that colony can be taken on slide and then cover slip is applied. But here the morphology of the fungi get distorted. Second slide culture. So on slide, the STA block is taken and on that the growth is done and then can be seen under microscope. Cellophane tape method. So the cellophane tape is applied on the agar block and then can be put on the slide and then growth is seen, colony is seen. So that is cellophane tape mount. Laboratory methods for identifying fungi. So morphological identification, rate of growth at 37 degree or room temperature 25 degrees Celsius. Color and morphology of the colony and its texture. Pigmentation on the obverse, that is front, and reverse, that is backside, whether diffusible or non-diffusible. Microscopic identification. So LPCB mount is done, that is lactophenol cotton glue. So in that lactic acid, phenol and cotton glue for the glue coloration. So in that can see hyphae that are septate or aseptate. So aseptate hyphae is in mucor, septate hyphae is in dermatophytes. Mycelium, vegetative or aerial, conidia or sporangium. Then other tests for identification of fungi, germ tube test in Candida, hair perforation test in dermatophytes, urease test, Cryptococcus, fermentation test, antifungal susceptibility test, antibody detection, antigen detection, cryptococcal antigen in CSF by latex agglutination, rapid diagnostic test, nucleic acid hybridization, polymerase chain reaction test. Hello students, let's study dermatophytes. So dermatophytes are superficial mycosis. It is group of related fungi capable of infecting keratinized tissues, skin, hair and nail. Species Trichophyton infects skin, hair and nail. Microsporum species infects skin and hair. Epidermophyton species infects skin and nail. Clinical manifestations, dermatophytosis or tinea or ringworm grow in centrifugal pattern in the stratum corneum, annular or ring-shaped pruritic scaly skin lesions with central clearing and raised edges. According to the location, body, trunk and limbs, it is called tinea corporis. In fact, feast the ribs between toes. It is called tinea pedis or athlete foot. Infection of the groin area, tinea pruris or jock itch. Beard and mustache area of the face, tinea barbi. Non-beard area of the face, tinea facie. The species Trichophyton, Macroconidia rare, thin-walled, smooth pencil shape, and Microconidia abundant. Microsporum, Macroconidia numerous, thick-walled, spindle shape, and Microconidia rare. Epidermophyton, numerous, smooth-walled club shape, Microconidia absent. Lab diagnosis of dermatophytosis, specimen collection, skin scraping, hair plucks, broken or scaly ones, and nail clipping from the active margin of the lesion. Hair should be plucked but not the cut. Cabbage mount or cauliflower white stain mount, thin septate hyaline hyphae with arthroconidia. Then culture on SDA, Sabrot's dextrose agar containing cyclohexamide, incubated at 26 to 28 degrees Celsius for 4 weeks. Potato dextrose agar, better sporulation. Identification by macroscopic appearance of the colony, rate of growth, texture, pigmentation, colony topography. Microscopic appearance by LPCB mount, lactophenol cotton blue tease mount. Hyphae and conidia are seen. So conidia, two types, microconidia and macroconidia. So microconidia small unicellular and macroconidia multicellular septate. Other test, hair perforation test, fungi pierce hair producing wedge-shaped perforations. Positive in tinea mentagrophyte, Trichophyton mentagrophytes and Microsporum canis. Urease test, Trichophyton mentagrophytes is urease positive.
Hope you find this session useful for your understanding and for your exams. Thank you.